later, there's Catherine for our lightning talk session. Awesome.
Yeah, hi.
Thanks, Catherine.
The next 30 minutes, we're gonna have 5 lightning talks. I just quickly introduced Sanchita Bhadra. Oh, I'm super sorry, Sanchita Bhadra from University of Texas, Austin. Sanchita, are you there?
Yes, hi Catherine, I'm here. Hi everyone.
Hi. All right. Ellie, can you share the screen? Sanchita?
Yes, I can share it. Start sharing.
So for everybody, please feel free to share your questions in the chat. We will have time for 1 or 2 in the end of the talk. And I will give you a quick reminder after when you just have 30 seconds left. So Sanchita, please, have the show.
Thank you, Catherine. Hi, everyone. So today I will briefly tell you about a new molecular biology reagent platform that we call Cellular reagents that is both low cost and locally sustainable. It has been developed by our team in the Ellington lab at UT Austin in collaboration with Jani's group at Cambridge University, as well as their partners in the Mboa lab in Cameroon and Hype Bio lab in Ghana. So what are cellular reagents? So cellular reagents are basically dried bacteria that are used directly as reagent packets without requiring purification of the protein that they carry. So the production process is deliberately minimalistic. Simply transform the appropriate E. coli strain with the expression construct and induce the protein production. Then collect the bacteria after the protein has been produced and distributed into either tubes or solid platforms such as filter paper discs, and dry the bacteria overnight in a box that contains some chemical desiccants. And this can be done at 37 degrees C. No cold chain is needed through the entire process. And once prepared, the cellular reagents can also be stored at ambient temperature for long durations. And for usage, simply the cellular reagent has to be rehydrated, usually in water, and directly added to standard reactions by simply switching out the typically pure reagent that is used in these processes. So in this manner, the cellular reagents can be used in a variety of pretty common molecular biology processes, such as PCR, as seen here in an agarose gel image, as well as we can run quantitative PCRs using cellular reagents. They can be used for isothermal reactions, as seen here in a LAMP assay for detection of SARS-CoV-2, either by an endpoint fluorescence readout or colorimetric lateral flow readout. They can be used for reverse transcription, as seen here in a RT-qPCR, as well as for DNA assembly processes. And in most cases, the performance is quite comparable to pure reagents, especially in nucleic acid amplification or diagnostic arenas. Cellular reagents are also locally sustainable, and this was shown in a very nice way by Jenny and her group. So they basically manufactured one of their open enzymes called OpenVent in UK, as well as subsequently in Cameroon and Ghana, and often using DIY bacterial incubators, as you can see here. So the setup needed to make these reagents is fairly simple, and we think it can be brought up in locally without investing a lot in infrastructure. And then as shown here on the right, these are some PCRs they executed with their OpenVent cellular reagents, and the amplicons can run, in this case, even up to 7.5 kb. So in conclusion, by simplifying the production process and eliminating the cold chain, we think that cellular reagents offer a low cost and locally sustainable alternative to commercial molecular biology reagents. Consequently, we think cellular reagents have the potential to enhance the affordability as well as accessibility to molecular and synthetic biology research and diagnostics, as well as further technology development. With that, I want to thank the funding agencies that supported this work, as well as all the audience here today for their attention. I probably zipped through fast, but I'm happy to answer questions.
Thank you, Sanchita. So if there are any questions, feel free to raise your hand and take the lead. If you have any questions later on, feel free to post them in the chat and we will address them to a later moment.